Stool in a human being is typically composed of about 300 to 1,000 different types of species. Testing stool is important for gut science research because it basically gives us insight into what microbes are dominant or deficient in particular individuals to understand in the context of what disease that may be associated with. My name is Dr. Deanna Gibson and I'm an assistant professor at the University of British Columbia on the Okanagan campus. Today we're going to be going through the process of collecting stool from a donor all the way to data analysis.
Hi, my name is Nishat and today I'll be showing you how to prep your sample when you poop for science. So this is our kit here. Inside our little styrofoam box is the tube that we'll need to collect our sample. We have a spatula to scoop up the poop. And of course, this process wouldn't be complete without some gloves to keep everything clean and not messy. We're gonna try. I am fairly well acquainted with fecal matter, given that I've spent about 5 years now exploring the wholesome, wonderful world of poop. I know about the bacteria that are there teeming with microbes. Our stool is. It's a wonderful place for microbes.
Hi, I'm Jacqueline. This is Deanna. And we're going to be doing a stool homogenization. So Deanna is currently pouring out our liquid nitrogen. So Deanna is adding liquid nitrogen into our insulated bowl in order to pre-chill the bowl so that we can add the sample that was so graciously donated by Nishat. Now that the bowl has chilled down, we're going to add our sample. I definitely have a new appreciation for the diversity of stool. I don't think I ever realized it came in so many colors. So we're just breaking it down into sort of more manageable pieces, breaking up any big chunks. Generally, for a stool sample of approximately 10 mils, it usually takes us about 10-15 minutes in order to get to this consistency. And so once this consistency has been reached, we transfer it into our tubes. 2 of our tubes are filled only a quarter of the way full. And so these ones are going to be used for extractions, for DNA extraction and for RNA extractions. I've just completed the extraction tubes. We're going to move them into our storage box and put them into the -80-degree freezer. Again, just we want to keep everything nice and cool.
Hi, my name is Candice, and today we're going to be doing DNA extractions from the homogenized stool. So first, we get the homogenized stool samples out of the -80-degree freezer. We're going to add a lysis buffer and a homogenization bead, which will break the stool up further so that we can extract the DNA. What we can see now is all the debris has gone to the bottom and we have a nice supernatant that we are now going to collect. We're going to separate out the supernatant and we're going to do several steps to clean it. And this whole process will take somewhere between 4 to 8 hours, depending on how many samples we have.
Hi, my name is Nijat. I'm doing the bioinformatics analysis for this whole process. So after the sequencing is done by the company, they sent us the data and I start analyzing this whole data. So this is the whole raw data, what it looks like. It consists of whole DNA sequences and it's long and it's very noisy. And we use other bioinformatics tools to do more visual representation of what the data would look like. And then our data will be analyzed in the form of visual representations like this.
As you can imagine, there's quite a bit of challenge when it comes to interpreting all the data that we gather. One of the ways we can look at microbiota data is using computer algorithm to reduce the entire community of a single individual and put it into a 3-dimensional or 2-dimensional plot where it shows the distance between someone's community compared to another individual, for example.
Pretty much we can understand every physiological response from stool, in my opinion. You can look at it and you can understand from its color whether there's something going wrong in your body or not. If my kids get sick, the first thing that I look at is their stool. I'll actually go and watch them poop and then examine it in the toilet because it can tell a lot about the physiological status of that person.